All right, welcome back. Today we're going to talk about speciation and further talk about evolution. Anytime we talk about evolution, a lot of times when we first introduce evolution, we're talking about really the big picture and how all organisms have really had to change over time because the environment has changed, and as competition increases and changes along with the environment changing, everything really has to change to kind of keep up. So when we're talking about that, really the big picture and talking about different species. We're really talking about macroevolution, talking about even if you were to talk about just a genus or a class or an order, we like to look at the whole animal kingdom and really discuss that when we talk about evolution. That's macroevolution, but once we start talking about a particular species, which is what we really get into as we move forward, we like to talk about one particular species, and we look at maybe a distinct feature that they have and how it's changed over time. When we're talking about the Galapagos finches and we're just kind of homing in on their beaks, that's looking at microevolution. And when we talk about microevolution, well, what's going on? Why is it that beak is changing over time? Well, it's the change in the allele frequency over time. The better alleles are going to increase in frequency over time as they are more fit for that environment. And like we've said before, the environment really dictates what the most fit allele is, or the most fit, really the most fit trait is, because it's not interacting necessarily with the allele, but it's interacting with the trait, not interacting with the genotype. It's really interacting with the phenotype, although the phenotype is dictated by the genotype. But we like to draw the line between the two because we have those heterozygotes where they have that hidden recessive allele in there. So not necessarily interacting specifically with the genotype or the allele, but it's really interacting. The environment is interacting with the phenotype. And when we try to think about how is it that we have one species or another species, and when do we see that these things are really splitting apart, or when does one species become two completely separate species? That's talking about speciation. So as much as when we look at the big picture, it's kind of easy to tell, hey, you know what? A dog is not the same species as a cat, or a human is not the same species as an earthworm. That's simple. But when we start to look at really, when we're looking at phylogenetic trees, we've looked at phylogenetic trees before, and we look at the nodes of the trees, right? We say, hey, you know what? If I have this phylogenetic tree and it branches off here, and then there's a branch off here, and this one branches off this way. Well, we're looking at these specific nodes. Well, this node is going to represent a species. So up until this point, it was the same species, and then it became two separate species. And then from there on, this species then became this species, and it became this species. And we see this speciation, this process of one species splitting into numerous species over time. But really, when you try to look at the nitty gritty, when does it exactly occur? It's very difficult for us to really, put our finger on it. So that's really what this is all about. We have these ideas of what different species are, but when we try to, as biologists, as scientists, we try to pinpoint an exact time that this happens, it's a lot more difficult than just saying, hey, you know what? You have a this type of bird and this type of bird, or you have a human and an earthworm, or anything like that. So speciation forms a conceptual bridge between microevolution and macroevolution. When we're looking at the bigger picture, the macroevolution, we're ultimately also going to dig deeper into the microevolution. That's really being looked at by understanding speciation, by the splits in the species. We all came from a single cellular organism, and then we continued to branch off into more complex organisms. And by us trying to look at, when did these branching offs occur when in the phylogenetic tree did we see that this became this and this became this, and they branched off into different organisms over time? That's all really looking at speciation. That's what's going to bring together that macroevolution with the microevolution. So species is a Latin word meaning a kind or an appearance, and that's really, we like to talk about species as being really the most specific term when we're talking about organisms. And we have more specific. You're going to hear about sometimes you hear about subspecies, or you'll hear about an ecomorph, and these are terms saying, we're looking at a species, and then they're all one type of species, but in that one species, there's different sects or different groups where we see differences. You think about things like humans, for example. Humans have different traits. If we wanted to separate humans based on something like race, right? If things never became homogenized the way they are now, where we have people of all different races living all over the world, and things were how they once were, you would have Asians in Asia, the we call them Mongoloids. You have your Negroid descendants that are going to be in the African region, and then you're going to have your Asian or your Caucasoid individual. I'm sorry, your Caucasian or your Caucasoid individuals. Your white individuals are going to be in Europe or more north. So if you want to create, they're all Homo sapiens, but you have these subsects of them, and that's where you're going to hear about subspecies or ecomorphs. And you can think about a race as, that could be kind of like a more a distinction that's more specific than species. But when we're talking about the animal kingdom and the way that we name species, we are always going to use the genus and the species. We've talked about that before. So species is really the kind, the most specific. So biologists, they like to compare the morphology, the physiology, the biochemistry, and now what we really are looking at is the DNA sequences, because that is really what's going to tell us if something is related, not related, how related are things. So once upon a time, when we looked at really the morphology, for the most part, how things looked, that, or the anatomy and the physiology, all of those things kind of led us in the wrong direction, because some things end up looking very similar to one another because of the environments that they might live in, and they might not be very related at all. And something else that doesn't look like them because they got separated more recently, they might be more related, even though they don't look anything alike, and something else does kind of look like it. So when we like to think about different species, as much as once upon a time we use morphology and physiology and even the biochemistry involved in a species, now we can really pinpoint things by looking at the DNA. But we like to group organisms by looking at all of these things. So with speciation, obviously you're thinking now, you know what, this is maybe more complex than I ever thought of it before. And with starting to dig deeper in speciation, you realize that, well, there's more to it than just saying, what, a sparrow is not the same as a finch, or a Homo sapien is not the same as a monkey, but or a chimpanzee. But there's different concepts involved with them, and we like to talk about different concepts. The big one that a lot of people like to use is this biological species concept, which is going to say that that a species is a group of populations, and the members have the potential to interbreed in nature, and when they interbreed, you're going to have these viable and fertile offspring. So what does this all mean? Well, it really means that a species is a species if it's going to breed with the whoever it's going to breed with. So if you have a group of say finches, and they all look very similar, but you have this group that only breeds in within itself and another group that breeds within itself. You might say, hey, you know what? They don't breed together. When they're put together, they're not going to breed with each other. So that makes it two separate species. Okay, so they don't breed together. So we just look at it as, if you're going to breed, if we put you together and you're going to breed, you are part of the same species. If we separate, if you are not going to breed, then you are going to be in a separate species. And that's a simple way of looking at it. Say, hey, you know what? This is how we are going to define if something is part of the same species or if it's a separate species. So gene flow between the populations holds a species together genetically by looking at this biological species concept. And you see that there's diversity within a species, and that's where it gets tricky with the morphology. And if you just keep it plain and simple by looking at the biological concept of speciation or of a species, you can say, you know what, as long as we put them together and they're going to breed, they're the same. So you could say, hey, you know what, all Homo sapiens, no matter how they look, the diversity in their appearance or their morphology doesn't matter because we put them together and they're going to breed. Now, with reproductive isolation, okay, we have this existence of biological factors or barriers that are going to impede two species from producing viable, fertile offspring. So when we have hybrids, they're going to be offspring that result from a mating between the different species. All right, we take two different species, they're going to mate and they are going to create hybrids. So now we see that it starts to get kind of wonky because now we have things that we are considering different species, and suddenly they found something else that they are going to breed with, but they are so different. You say to yourself, wait a second, are they the same species? Are they different species? What's going on now? Because now this idea of, oh, let's just see if they're going to breed together or not is not the answer that we're looking for. So reproductive isolation can be classified by whether factors act before or after fertilization. So we look at different factors that involve if species are going to be able to reproduce together, and we draw this line down and it cuts right through fertilization. So there's these pre-zygotic barriers, things that are going to happen before fertilization, and then things that happen after fertilization. So there's various reasons why things cannot interbreed, why you're not necessarily going to, even if so you're going to have ones where they're not going to be able to breed, and then other ones where they can breed, but are they going to make an offspring that's also going to breed, or an offspring that's going to be viable? So you have pre-zygotic barriers that are going to block fertilization from occurring. It can't happen at all, impeding different species from attempting to mate, preventing the successful completion of mating, hindering fertilization if mating is successful. So even if you're able to mate with this organ or this individual, I should say, is fertilization on the cellular level, going the sperm and the egg, going to occur? So you have habitat isolation where two species encounter each other rarely or not at all because they occupy these different habitats. So if you are in two different habitats, you never run into each other. Well, that is going to be a physical barrier that is going to stop you from being able to reproduce with that species or that individual, I should say. Then you have temporal isolation, where species that breed at different times of the day, different seasons, different years, they're not going to mix their gametes. So you would eventually consider them to be different species if you see that, you know what, they originally are the same species, but you have this group doesn't doesn't breed in the morning, and this other one that only breeds in the morning, or something like that, or ones that like to reproduce in the springtime and other ones that like to reproduce in the fall, just for an example. So there's also behavioral isolation, where courtship rituals are going to get in the way. We see a lot of times with birds, they have different calls and different ways of courting individuals. So if you are initially part of a species, and then you're going to have a group that start courting one way and another group that start courting another way, then you're going to create this behavioral isolation between the two. And then you have mechanical isolation, where morphological differences can prevent successful completion of mating. The parts don't come together, or something doesn't kind of work in order to, or the anatomy doesn't really fit together for the two to reproduce together. You have gametic isolation, where the gametes, the sperm of one species, isn't able to fertilize the eggs of another species. And then once we get past that, we start talking about post-zygotic barriers. All right, so you talk about fertilization, all right, preventing fertilization from happening. Those are our pre-zygotic barriers. Then you're gonna have your post-zygotic barriers, where it's going to prevent the zygote that's formed, the fertilized egg, from developing into a viable fertile adult. So these things include, the post-zygotic barriers are going to be, reduced hybrid viability. So when you create a hybrid of the two different groups or species, it's not going to be viable. If it is viable, it's not going to be fertile in order to make more of those hybrids, and then hybrid breakdown as well. So you have reduced viability, where genes of different parent species may interact and impair the hybrid's development or survival in its environment. So it's not a viable hybrid that is created. Obviously, reduced fertility. So even if the hybrids are created and they are healthy, that they actually are then sterile, meaning they can't then reproduce. So you have a great example is a mule. So down here you see this is a mule. And what is a mule? You might hear this sometimes, people talk about them kind of using the same term. Mule, donkey sounds like the same thing to people. Well, really, it's two separate things. A mule is something that we like to create in order to help us to plow fields, in order and to use them as pack animals to carry stuff for us. So we actually create a mule, and the way that you do that is that you take a male donkey. So here you have your male donkey, all right, and you have a female horse, and you're going to mate the two, and now we're going to artificially inseminate the the horse. And then when you mix the donkey sperm and the female the horse egg, then you're going to create this mule. And a donkey has 62 chromosomes, and a horse has 64 chromosomes. And believe it or not, they actually come together, and they're actually the sperm is actually able to fertilize the egg, and it works rather readily. And we're going to see that a mule ends up having 63 chromosomes. And believe it or not, it works just fine. But what happens is, when you try to take a mule, you are actually not going to be able to reproduce with that mule. You're not going to create a viable offspring. So they are sterile, which isn't the end of the world. We can just do this all over again, where we take a donkey and we take a horse and we create a mule. And we like to do this because what you actually have is, you have the strength of a horse, strength of the horse, but you have the intelligence as well as the endurance of a donkey. So we kind of have the best of the worlds, of both worlds, and that's how we create our mule. But it is not going to make, all right, you're going to have a viable offspring, but it's going to be a sterile offspring. And then you also have hybrid breakdown, where some first generation hybrids are fertile, but when they mate with each other, or with a parent species or a non-hybrid species, the offspring of the next generation are feeble, or a lot of times they're going to be sterile. So these are the different post-zygotic reasons that reproduction cannot occur. So there are some limitations, as you're obviously thinking about, of the biological species concept that we're talking about. So the biological species concept, where we're talking about, do they mate or do they not mate? Do they interbreed with each other? So this concept cannot be applied to, when we look at fossils, and really we can't use this for asexually reproducing organisms. So all prokaryotes, we can't decipher between different species by using this biological species concept. So that's a pitfall to this biological species concept. The biological species concept also emphasizes absence of gene flow. And then you also have, as much as we like to use that example of the mule with the donkey and the horse, there are examples where you have very distinct species, just like we think of a donkey and a horse being distinct species that are going to make that mule. Well, a grizzly bear actually and a polar bear, they can mate and they make a growler bear. And believe it or not, growler bears are fertile. So this kind of throws a whole wrench at you. You think that with this you have a grizzly bear, which is one unique species, and a polar bear, which is a unique species of its own, two separate species completely. But they're actually able to mate with each other, and once they mate, not only do they make a viable offspring, but they also make an offspring that is not sterile, is able to reproduce. It is a fertile, viable offspring. So that kind of throws a monkey wrench in our whole idea of the biological concept of speciation. So other definitions of species, we talk about the morphological species concept, where and this is really how they used to do things before DNA was involved. They like to look at, hey, what do things look at? Looking at the structural species, and it applies to sexual and asexual species. So it's nice now we can take care of our prokaryotes when we want to. But it really relies on subjective criteria, and as scientists, biologists, we like to have objective criteria, objective data. We like to look at something where it's not really up for opinion. So subjective data is something that can be really different from one organism to the next, right? We always talk about subjective data as in pain. So when you go to your doctor, they are always trying to look for objective data. They want to know what your temperature is. They want to know what your blood pressure is. Those are definitive numbers, objective data. If I ask you your pain, well, pain is very different from one individual to another individual, and that's going to be subjective. So I might think of a certain amount of pain as being a 10, and I don't really have a high threshold, and then someone else might think of it at that same amount of pain as a 1. So that is very subjective. And when we're talking about morphological species concept, it really comes down to a lot of it is very subjective, and we just don't like that in science. So apart from that, you have your morphological, you're going to have your biological, and then you have your ecological species concept, which is going to define species in terms of the niche that they fill. All right, so in the ecological role, in their ecosystem, in their environment, what role do they play? And this applies to both sexual and asexual reproducing species, and emphasizes the role of disruptive selection that we've talked about. But many species definitions have been proposed. The usefulness of each depends on the situation and the research questions being asked. And the issue with the ecological species concept, the niche that they roll, is that we have these things that we like to call ecomorphs. And ecomorphs are actually the same species. Same species. Terrible handwriting, I apologize. Same species. So ecomorphs are they are the same species, but they actually act very differently in depending on which role or which niche that they are in or niche that they are in. So same species, excuse me, but they are ecomorphs. So that kind of throws a monkey wrench in the ecological species concept. And within speciations, we like to talk about two specific types of speciation that occurs, two different ways that you can ultimately create two different species. You have allopatric speciation, which is probably the easier concept to understand, and then you have your sympatric speciation as well. So in allopatric speciation, you're going to see gene flow is going to be interrupted or reduced when a population is divided into geographically isolated subpopulations. So when there's some kind of divide in these in a population of organisms, and then over time, from being separated, you're going to evolve separately. You're going to have that microevolution going on in the two different populations. So, for example, you have the flightless cormorant of the Galapagos Islands, and these are birds that don't fly, and they originated from a flying species, and over time they stayed on the islands of the Galapagos, and as they kind of hung out there, they slowly started to change, and they became their own distinct species that was very different from the mainland flying species, because they started to adapt to a completely different environment than the ones that were living on the mainland. Separated populations may evolve independently through mutation, natural selection, genetic drift. Reproductive isolation may arise as a byproduct of genetic divergence. You have examples like isolated populations of mosquito fish have become reproductively isolated as a result of selection under different levels of predation. And then the flip side is this idea of sympatric speciation. So allopatric, easy to think about, hey, these populations are being separated somehow. All right, maybe there was some earthquake and there was some break in the land, and now they weren't able to meet up with each other. Maybe there was some flooding and they got separated. Maybe there was some of the population got washed away and went to an island, like with the Galapagos, and the mainland ones were separated. Simple. But then you have sympatric speciation, where this occurs in populations that live in the same geographic area. They are not separated geographically like in allopatric speciation. They're still living side by side, but for whatever reason, you're going to see that they create separate species within that same geographical area. And some factors are some polyploidy. You have sexual selection, and then you have habitat differentiation. So here you go. Here's a nice diagram of your allopatric, where somewhere along the way they maybe the water went down and these got separated from each other. Okay, and now these ones are evolving in this specific ecosystem or this environment, and these are evolving in this one. Then you have sympatric, where they're still able to reproduce with each other, but for a specific reason they are starting to form separate species. So polyploidy is the presence of extra sets of chromosomes due to an accident in cell division. So much more common in plants than in animals, but we do see it in animals, and it can produce new biological species in these same countries, or sympatry within a sympatry within a single generation. So you have an autopolyploidy, which is an individual with more than two chromosome sets. And then the offspring resulting from mating between the polyploids and the diploids usually have reduced fertility. So here you have your diploid cell, and what do we want to do? Well, we want to then make another diploid cell, but what happens is because of an error, we create these tetraploid cells. So instead of creating more 2N cells, we first make a 4N cell, and then the 2N cells are going to make 4N cells again. So now that's an issue. That is polyploidy, because you are now having extra sets of chromosomes. We're only supposed to have our 2N cells. Now you are making these 4N cells. And allopolyploidy is going to be a species with multiple sets of chromosomes derived from different species. So they can successfully mate with each other, but cannot interbreed with either parent species. So here they show you, you have this set where you're going to end up with their 2N is 6 and their 2N is actually 4, and then their gamete's going to have 3, and their gamete's going to have 2, and that you're going to end up with 5. But then in the end, you're not going to create you're going to create these really different species from this polyploidy. So many important crops like oats, cotton, potatoes, tobacco, wheat, they're all polyploids, and they do just fine. Plant geneticists can produce new polyploid agricultural species using chemicals to induce these errors in these chromosomal errors in reproduction and meiosis or cell division. So with sexual selection, that can actually drive sympatric speciation, like I said, where you're going to still be together, but for whatever reason you have certain groups that are going to mate with certain ones. An example, different colors has contributed to speciation in the cichlid fish in Lake Victoria. And you have habitat differentiation where sympatric species can also result from the appearance of new ecological niches or niches. So, for example, populations of the North American maggot fly can live on native hawthorn trees as well as more recently introduced apple trees. And then the flies that use different host species experience both habitat and temporal isolation. So now you have these two different populations. One population of maggot flies that are going to like to be on the hawthorn trees, and then the maggot flies that are like to be on the apple trees. And over time, that's where you see the sympatric speciation due to the different habitats that they're within their habitat that they're going to live in. So it can occur rapidly or slowly, the speciation, and results from changes in few or many genes. So many questions really remain concerning how long it takes for speciations to form. And we see that the rate of speciation can be studied using fossil record, morphological data, or molecular data, and it really depends on each specific scenario. Sometimes it happens quick, sometimes it happens slow, and we like to look at different patterns of this speciation. So for one, you have this punctuated equilibria or a punctuated equilibrium that we like to talk about, where it describes that it's not always going to be this gradual thing. So the fossil record includes examples of species that appear suddenly, persist unchanged for some time, and then disappear. And that's our punctuated equilibria, where you're going to see that you have these times of stasis, and then it's going to be punctuated by sudden change, drastic change. So this model contrasts with the gradual change in species over time. So we like to always talk about this gradual change over time. It looks like this, right? You have this one species, and then it starts to look a little bit different, and then it becomes more different looking and more different looking, and over time, as that's happening, this one's looking even more different looking and even more different looking. But we see in punctuated equilibrium, you have this one, and maybe for a long time you just have this one, and then suddenly you have this drastic change, and then this drastic change can go on for a very long time, and then there could be another drastic change. So there's two different models that we talk about when we're talking about speciation. Keep these things in mind as we move forward and we continue to talk more about the ecosystems along with what we've learned about genetics and evolution.